attention. It's about 7:58. I'll just start the introductions, and then at 8 a.m. We'll start the first presentation. So good morning, everyone, for those in the U.S., and good afternoon to everyone in the U.K. Today we'll be having two presentations. The first one will be Reactome, and the second one will be MyGene.info. Today's presentations will run till 9:30 Pacific time to include more questions and discussion. Each presentation slot will be about 45 minutes long. Just know that if you can't stay for the entire length of the presentation, the whole conference will be recorded and can be accessed online. And just a quick reminder, if anyone has questions during the presentation, you may type out your question in the message box, and the presenter will answer when possible. Any questions? All right, if not, I'll introduce our first presenter. So our first presentation will be presented by Antonio Fabregat. Antonio holds a bachelor's degree in computer science and a master's degree in artificial intelligence. He is currently a software engineer at EBI and has worked there for 4 years. Antonio will be presenting about Reactome, which is a free, open source, curated, and peer-reviewed knowledge base of biomolecular pathways. One of its goals is to provide intuitive bioinformatics tools for the visualization, interpretation, and analysis of pathway knowledge to support basic research, genome analysis, modeling, systems biology, and education. Without further ado, allow me to introduce Antonio. Go ahead, Antonio.
Okay, thank you very much. Well, so today we are going to present a little bit what is Reactome and how data gets into Reactome and why the reason we have a database like Reactome, and then later on one of our tools on top of this data that is a web application. So, and it also have a server side for the data analysis and so on, but we will see every part in a few minutes. So in biology, when you find just the publication, the paper, human being can just read and go through all the information, and for the human being that is easy to understand, or well, if you are an expert in the field, then it's easier to understand. But for a computer, just text as it is in the paper, maybe could be a little bit difficult to parse and understand. So in Reactome, they are experts on the field, and what they do is just, well, it's a very important work. So they just go through all these papers and all this information, and then they figure out the meaning of this, and they curate this data into the Reactome database using one the Reactome curator tool, but that's out of the scope of this presentation. So later on Reactome, once this data is modeled and is being introduced in the database, people can just access to it in several different ways, either through diagrams. So we have some sort of diagrams that are visual for the people, but you also have access to the raw data, so that we're accessing directly to the database or accessing through a RESTful service and so on. Okay, so for example, the aim of Reactome is just to model reactions. So in reactions, what you have is inputs, you have outputs, and then you have how is this called? Activity. I forgot the name. Catalyst. Catalyst. Yeah. And you have catalyst, and well, in reaction. In Reactome, at the end, the curators, what they are doing is curating these reactions. So every reaction the scope of every reaction happens in different parts of the cell. So in this case, we have here a reaction where we have one part of it happens in the compartment platelet dense tubular network lumen, and then part of the information that is in this compartment goes out of it after the reaction happens. And yeah, and then every of these reactions we identify with a stable identifier that remains remains stable through different releases. We have several types of reactions. So the curators are curating like the building reactions, phosphorylation, the dimerization reactions, and every one of those has a specific meanings in biology. And for the experts on the field, so it's one of them. So sometimes what you have is proteins that they merge together to just come up with a complex, or sometimes you have the input is a complex and then it gets split into different proteins, or you have just one physical entity that gets transformed somehow into another one just by some internal chemical reaction. Okay, so once we have a reaction created, if it happens that the output of one reaction is at the same time the input of another reaction, or it could happen that the output of one reaction plays the role of the catalyst in a different reaction, we can then join all of these reactions in a kind of chain, and the result would be a pathway. So a pathway could be explained as the concatenation of different reactions, which can just happen because the product of one is the input of the other. Okay, so these reactions are also called events. So for us, when several events are all put together, this is called the pathway, and the pathway is at the same time an event. So if we put all this together in an ontology-like structure, what we end up having is grouping these reactions. For example, can you see, when I move the mouse, can you see the pointer on your screen?
Yes, we can see it.
Okay. So here, for example, in this tree, in this ontology-like hierarchy, you see that there is a pathway and there is a reaction. So on the reaction, we have two types of pathways. Would you please, I don't know. Please mute yourselves if you're not speaking. Okay, now it's much better. So here we have that all of them are part of the same pathway, and then when you can also group pathways and at the same time in a super pathway, and so on until we get into the higher level of this hierarchy we call the top level pathways. And well, something I specific is that some reactions can happen at the same time in several locations, but that's out of the scope of this talk. So,
Antonio, could you repeat what you said earlier during the feedback? We couldn't hear you.
okay, start on the slide again. Okay, I start again then. Okay, so this is like you have to start. Okay, so in this slide, what we show is an ontology-like hierarchy where we have a few pathways in Reactome and also some reaction. And what I was saying is the reactions are at the end of this hierarchy, of this ontology-like hierarchy, and you can group several reactions into one pathway. And then at the same time, pathways can be grouped in super pathways or wrappers. And you can do that going up on the hierarchy until you reach the top level pathway. Okay. Yeah, and what I was saying that it happens that sometimes you have reactions or pathways that can be at the same time in several locations, but that's out of the scope of this presentation. So if you connect to Reactome, to our website, then you have the access to many tools and documentation and files that you can download and explanations and so on. But today our aim is focusing on the pathway portal. That the pathway portal is a web tool suite that is built around different tools in Reactome, which integrates biological pathways and allows browsing and analyzing your data and displaying these results in a graphical way. So the pathway portal is still under development, so we try to add more stuff depending on our users' requirements or what is needed at any time. And then this web tool suite contains several modules. So here we have that the main module that one of the main modules that we have is this pathways hierarchy navigation that you saw in a couple of slides before. And we also have So can you hear me now? Yes, of course. Okay, so we also have a pathway diagram display and a details panel. And in the details panel we will see that we have different tabs, but I better go to this slide. So if you can see the mouse, I will show you. This would be the hierarchy, the pathways hierarchy module. Then we have here in the main module, we have the diagram, and in the future we are going to include here the fireworks. If you connect now online, the fireworks is not yet available, but it will be in a while. Then in the details module, we have several panels or several tabs, which are the overview, the molecule tabs, structures, expression, analysis, processes, and downloads. And in every one of them, we are representing different data coming from different resources. So, well, in the overview tab, that is the one that I was showing in this slide, what we show is data coming from the Reactome database directly. This is the data directly curated by our curator, which contains fields that like the summation or the input of the reaction, or well, the specific fields for every kind of object that you have selected. Then the second tab would be the molecules tab. This is partly, well, this tab is also coming from data directly curated by our curators. But this is kind of interactive with the pathway browser. So when you select, for example, here a reaction, then the molecules tab will tell you that in the pathway where you are, there are 122 molecules. 112 of them are proteins, and in these reactions, there are 3. In this specific reaction that you have selected in the diagram, the proteins playing a role here would be these 3. Okay, this is automatically changing depending on the entities selected in the pathway browser at any time. Okay, if you expand one of these, okay, then in the diagram we can show interactors. In this case, we are showing data coming from the intact cyclic service. Then here in this case, I selected just 3 proteins in the diagram where I chose to see the interactors, and as you can see in the diagram, in the slide, we have several. For example, this protein is interacting with the 3 of the proteins that I selected, so you have direct links. In the tool, if you double-click the edge, then you go to the intact page that has information about this interaction. Then at the same time, here in the molecules tab, you can also expand, for example, in this case is this protein, the one selected in blue in the diagram, and you can see cross-references to other databases. Okay, if we go to the structures tab, for a selected entity, in this case we have selected a small molecule, we are showing the structure. So for the small molecules, we are connecting directly to Kevin, and then we show the Kevin name and secondary identifiers, and the most important bit that this would be the chemical structure in the picture. Then if we select a protein, in this case we have selected this protein over here, in the structures tab what shows us is the protein structure. This data is coming from the PDB and is fed as the KV is fed in real time, so we don't keep this information. This is retrieved by the pathway portal automatically. And then you have the option of see other structures. So by default, it shows you the best one, the one that the PDB says that is the best one, and then you can expand and see several other structures that have different coverage or different resolution or so on. Okay, if we select a complex instead, what we see is the structures for every protein that is contained in the complex. In case the PDB doesn't have the information for one of them, then a message that no structures found is shown. And the same than before, you can expand this all other structures, and then you get more detailed information. Okay. In a few cases that we are still getting into this kind of data, we also have RIA connections. So if you select a reaction, then what you see here is the chemical reaction represented in RIA associated to this reaction. There are only a few of these RIA reactions modeled for the time being, but we are going to try to increment this number in the future. Okay, now moving to the fourth tab, we have the expression tab, where we connect to the Gene Expression Atlas database, and this widget is provided directly by them. It's a Bio.js widget, and in this widget, for the selected diagram, in this case, we see all the genes that are in this diagram, in this pathway, and the level of expression for each one of them. This widget is interactive with the rest of the table. In the slide it's not shown, but when you move the mouse on top of it, different columns, different rows, the human body is highlighted, and the other way around. So if you select the human body, then the rows are also highlighted. And this information is not stored in Reactome either, so this information is directly retrieved from Gene Expression database or resource. Okay. Then for the next step, first I need to explain a bit the Reactome Pathway Analysis Service, but some of you have already worked with it. So the current, and the analysis service is a RESTful service that provides analysis against the Reactome pathways, and it uses the entities main identifiers, entities secondary identifiers, cross-references, and other identifiers, so the final user doesn't need to come with the uniprot identifiers or the sample identifiers. We are cross-referencing to a number of different resources, and if your identifiers are one of these resources, then the analysis set will map these identifiers to the main Reactome identifiers and will provide you with the result and also with the mapping. Okay, for the time being, we are only taking into account the data directly curated in Reactome, but in the future, we are planning to improve this analysis service by including protein-protein interaction from impact from the Cyclic service, but this is still work to be done. Okay, so in the Pathway browser, we have an analysis tool, let's say a client for this analysis service, which allows the user to select either a file from the computer or just paste as a set of identifiers that are the target of the analysis. And once the analyze button is clicked, the result comes into gets integrated directly into the Pathway browser. Is overlaid in the hierarchy. Is overlaid so those pathways that have been hit with the user sample are have the FDR and the number of entities found in this pathway, again the number of total entities in the pathway, and we also show the result in the analysis tab. So this result, oops, sorry, this result shows the by default it's ordered by the p-value, so the most significant pathways appear on the first page and on top of the table, and by selecting the rows in this panel, in this table, the diagram gets selected, and then the pathway gets selected, and then the diagram is shown in the main module, in the diagram viewer, and the overlay and it overlays the analysis result on top of the diagram. So in this case, we perform another representation, and then we use yellow for those entities that has been hit, and in some cases you see that there are boxes that only have the box color. That's because it's a complex, and what that is telling you is that only 50% of the proteins that are in this complex have been hit by the user data. Okay. So, well, the results can also, I say it now, the results for the final user can also be downloaded by clicking on the result button, and the same for the mapping. Okay. In this case, what we are showing here is that if you see, the user selects a sub-pathway, then we see the different reactions that are part of this pathway selected in the diagram with the overlay of the result, and at the same time, for those reactions that have been hit by the user sample, a yellow halo appears next to them. So it's quite easy to identify which are the reactions. From a significant pathway, it's quite easy to see the reactions that have been hit by the user sample. Okay. So in case we use expression data, so the user submits a set of identifiers but the identifier will have one or more columns with expression data, so kind of time series. Then in this case, the hierarchy and the analysis there, the table there behave exactly as I explained before, but for the overlay, what you see then is the expression values going from the lower expression value to the higher expression value, and also if there is a time series in the data, these controls that appear in the diagram allow the user to move from one column to the next, or from one item on the series to the next one, and so on. Okay. This is work ongoing. It's not finished yet, but it's in a very good status for the moment. Is the fireworks. So the fireworks is a genome-wide hierarchical visualization of the reactome pathways, and the idea is just to have a map filling the space, the variable space in the plane, to show these pathways and the parent-child relationship of them. So the aim is to visualize, the final aim is to overlay the analysis results on top of this fireworks view to allow the user to have a very quick first glance and a rapid zooming on the rapid zooming in on the interesting results. So in those pathways that are very significant, and yeah, and the idea is that the user can just see not as a single pathway, but a set of pathways in a certain area of these fireworks that are of interest. So the alpha version is already integrated with the analysis service. So for the moment, we can overlay the analysis result data. But the time that I prepared the presentation, only overrepresentation was shown. So in these slides, I haven't included any expression data or species comparison, but that's something that will happen in the future. So this is how the fireworks look like. So it would be integrated in the main panel of the pathway portal, and it's connected with the, so if you move the mouse over the hierarchy tree, the corresponding pathways will pop up a little bit, so the name will be highlighted, and then if you select any pathway in the fireworks, this will be propagated into the hierarchy, and at the same time you can see all the information that I've shown you before in the different tabs related to the pathway. Okay. So by zooming in a little bit, well, the common thing is that the thumbnail here shows you the area of the fireworks that we are zooming right now. And by selecting what I told you, if you select one pathway here, then this selection gets shown here in the hierarchy tree. The citric acid cycle is the one selected, and you can see in the fireworks that has 1, 2, and 3 parents, and those parents in the hierarchy tree are these 3 pathways that are highlighted in blue. Okay. Well, in this view, we only show the name of the selected pathway and the name of the top-level pathway associated to this citric acid cycle. But by zooming in, keeping zooming in, the granularity changes, and then you can see the name of all the nodes that are shown. Right now we see that the part of the firewalls that we are showing on the screen is this small bit here. And the idea is that once you get into a region of interest, then you start displaying more information related to this smaller space. This would be the result of an over-representation analysis. We have taken an example data, and what you see here is the fireworks now has several colors on it. So going a little bit quickly through the different colors, those that are fade out mean that the user sample hasn't hit at all in these pathways. Those that have a kind of pink color means that the user sample has hit these pathways, but this pathway is not significant for the user data. And then we have now pathways which color goes from brown to orange. So brown would be the p-value being 0.05, which is quite a nice p-value, but the best p-value is 0. So if 0 tells you that you have found, so this pathway is very significant because the proteins that you have found in these pathways is not by chance. Okay. So by seeing now, or having a look to the firewalls, what we see is that the user sample was hitting very nicely in the signal transduction, and it's also related with immune system, hemostasis, and developmental biology that in somehow are also related with this signal transduction pathway. At the same time, and maybe it's of the user interest, could be the cell cycle, because it has some branches which are very nicely hit. The same for DNA repair, and a few pathways here at the end of the metabolism hierarchy. Okay. Then I can move on by selecting. What I wanted to show in this slide is one of the in the firewalls. If you select one pathway of the hierarchy of the view, then the legend gets highlighted in blue. Well, gets a blue line where representing the level of significance of this pathway. So by hovering another one with the mouse without clicking, it shows a yellow line. So in this case, you would see that the GRB2 events, so the pathway GRB2 events in EGFR signaling has better significance, or better is more significant, so has better p-value than the nuclear events kinase and transcription factor activation. So, and that is, it's just a way of allowing the user to quickly compare two pathways by using the firewall. Something that I didn't mention, but it's important for us, or it was one of our aims, is that from one release to another, these firewalls, of course, will change because new pathways are likely to be added into the Reactome pathway hierarchy. But by doing that, we don't want to change the layout of the previous pathway. So in case there are inclusions, we want to maintain as much as possible the same layout. So user can just compare data not only or results not only for one Reactome release, but across different Reactome releases. And we also have the firewalls pathway, the firewalls view for different species. Keeping every orthology pathway would be in the same place as the normal pathway, or as the curated pathway is in the main view. If we have time later on, I will show you in the pathway browser in one local installation, so you will see how this firewall changes across the species, because I didn't include that in the slides. Okay, so something that we took into account. Let's see if I have forgotten something here. No. Okay, so something that we had into account when we were developing part of the modules of the pathway portal is the interconnectivity with other resources. So for us, it's very important to have users coming to our pathway portal and sending their data and using our web client as much as they like, but we are also interested on people using Reactome for their pipelines or for their web applications, so they can just analyze their data against our database and later on just connect to us to display the results, or just download the results through the RESTful services. That is something that is both possibilities are available. So currently, that we know, there are 3 resources already connecting to Reactome, that is Sprite, Blueprint, and COPAK-AV. The approach that these three resources has followed is quite the same. So the client connects to the resource. So the user, the final user, connects to the resource, either Drive, Blueprint, Copacabl, another resource, and then the user just requests a data analysis of some data hosted in any of these resources. So the idea is that the user is connected with his computer directly to, for example, Copacabl, and then Copacabl has a pool of data that for the user is interested. The user is interested to analyze against the Reactome analysis service. So what happens is that these two servers will get in touch, and for example, if we have 10 or more megabytes of data, this data doesn't really need to go to the client. So this data can be directly shared between the two servers, and the client would receive the final result. And once this data is analyzed and has a token associated, then the client can connect to the Reactome client, so to the Reactome web application or the pathway portal, to see the result overlaid with this that we have shown you. For example, how it works in Pride is that I put the Pride example because it's the one that I could easily access. So here we have a view of what the Pride archive is, and what they have is Pride is a repository of proteomics data. So people analyzing some tissues, they have a result, and then they send this information into Pride. Pride keeps the information, and now they have offered at this interface where you can just, by clicking in a button, you can take a bunch of identifiers for a certain project and then analyze against Reactome. In this case, this project, the aim of this project, it was to the identification of biomarkers for the histochemical diagnosis. Okay, I don't read the rest. And once this was analyzed and the view and the button was clicked, this is not available online, but I could have done it in my computer. So this is the result overlaid in the firewalls, and what we see is that the pathways that have been hit in Reactome, as a first glance here, as a first field glance, is that we see that the result is what we expected. So the immune system, well, the signal transduction, I don't know whether that can be part of the aim of this study, but the immune system, gene expression, metabolism of proteins, and the cell cycle is what the biologists would have expected to see with more significance once the data is analyzed in Reactome. So this is just a use case of the interconnection between these different services. With this very same data, we can just, the user can just select a specific pathway in the fireworks, and it will automatically open the diagram, and the result for this specific diagram will be overlaid. So they can open the pathway to display the diagram, and then the result will be overlaid on top of this view. So that was the explanation about what the pathway portal is doing to the moment. Okay, this well, thanks to all these people are part of Reactome. Reactome, I didn't say at the beginning, but we have people in the OICR in Toronto, here at the EBI, and also in the New York Langone University, Langone Medical Center. So thank you for your attention, and I would be happy to take any questions.
Thank you, Antonio.
Okay, I understand that nobody has questions, or is Victor?
This is Vincent. That was a great presentation. I have one question. This is more of a technical question. What type of technologies do you use to create the API for Reactome?
Okay, that is a RESTful service.
Yeah.
And the technology we use is Spring MVC, that is Java framework.
Sorry, can you repeat that?
Okay, the technology we use is a RESTful service, we have generated a RESTful service, and the technology is Spring MVC, that is a Java framework.
Okay. Thank you.
Hi, this is Chunlei from Scripps. I have a question. So, so the data stored in Reactome, like pathway or the reactions, where's the data source for those pathway and reaction data? Are they from the, say, like the curation or from other database? Something, yeah.
So this is Henning chipping in. So Antonio put up the next slide, which shows exactly how this happens. So all the data in Reactome for the human part, for the human pathways, is manually curated in a fairly in a fairly detailed way. We recruit an external expert for every pathway we want to curate. The expert, and then we ask the expert to do an initial description of the pathway, which often is a handwritten diagram and this pile of review articles. Then the curator will go through this material, will go for every fact where possible, for every reaction, go back to original experimental papers. So normally assertions in Reactome are not based on just the review paper, but really on the original experimental papers, which especially for, let's say, enzymatic reactions which have been worked on in the 60s is not exactly easy. We will, where that's not possible, we will explicitly mark this with so-called black box reactions. Then the curator goes back to the expert, says, are you happy with the representation in the database? And then we ask another external reviewer for a review. And only if both experts are happy, then the pathway will be released. So it's really very much like a scientific written manuscript review of the pathway, but in electronically accessible form. And of course, it is being maintained, so we do updates to proteins and things like this, so we ensure consistency over time.
I see. That's perfect.
This is for human. And then, next slide, yes. For the other 20 species in Reactome, we do orthology-based inference projection, so that when all the involved proteins in a reaction have a close homologue in the target species, then the reaction is projected to this target species, and otherwise it isn't. And of course this works fairly well from human to mouse, but of course it doesn't work well from human to, let's say, Arabidopsis. And that's it.
I see. Thanks. How how often the data be updated? Like is a fixed update schedule, or is it or maybe just continuously updating?
So there are two different levels. So the data itself, in terms of updating cross-references and the orthology projection, everything that happens automatic, sort of data maintenance that happens with every release, so every 3 months. And the content update happens in principle every 2 years. We aim to touch every pathway at least every 2 years. In practice, that is varying wildly between fairly frequent for pathways where there's a lot of new developments, or where we have local expertise, and it might be significantly longer than 2 years in some cases where not much is happening in that domain anymore, or simply we don't have the local expertise. But normally the target is at least every 2 years. One thing which is probably important to mention is Reactome is aiming to represent more or less textbook knowledge. So we on purpose stay back a bit from the bleeding edge of science, and let's say if there's a very new controversial area, then we will tend to not include this, but we will wait until the dust has settled a bit and there's really a consensus.
Great, thanks.
A short word probably on the coverage. So I think Antonio mentioned the coverage overall of Reactome. For a long time, that was the problem because with this complicated but very high quality curation process, we have had for a long time low coverage. But now, having reached much more than 7,000 Unicode proteins and significantly more in terms of isoforms, disease variants, and so on, we have now covered, I would say, all the major pathways, and we now see that we are getting into areas where it's getting more difficult to add new proteins because we've covered all the major areas. And we reckon that there's anyway only any knowledge, any functional knowledge which makes sense to put into a pathway for about 11 to 12,000 human proteins. For the rest, there's simply nothing known. So there's no point in putting it into a pathway. So we are much closer than completion than you might think if you compare 7,000, which we are touching now, to the 21 or so thousand genes, which you might think need to be touched. But for a lot of these, just there is nothing to do. But one part of why we are keen on including the interaction data is to increase the scope, and via the functional interactions of proteins which are annotated in Reactome, we aim to sort of increase coverage and be able to pull in data which we don't have annotated in Reactome directly. Of course, this will be appropriate to fact because it's fundamentally different from the carefully measured.
Comparing the results that for the significance, for example, it's quite difficult because we have to find also how to represent this kind of results because that could be
I suspect we will simply have a button include interactions in the analysis or exclude them. And then we can just try one step. Okay.
Maybe, yeah, so I was thinking I can try to show one of the give me 30 seconds that the Tomcat starts, and then I can show you a live demo in my local installation of Reactome. So we can see a little bit the behavior of the fireworks that I show you, saying that the granularity changes depending on the level of fruit. See a little bit how the overlay, how the analysis result overlay gets on top of the fireworks view, and then see a little bit how the pathway portal behaves.
So one thing to mention, Antonio didn't mention this in detail in the talk. We have invested a lot into the analysis, in the pathway analysis software architecture in the last year, and have changed, completely re-engineered it. Before, if you would send 5,000 proteins or genes to the pathway analysis, it would take between 5 minutes and 15 minutes or just die on the way. And now you can send the entire human genome if you want, and it will provide you an answer within seconds. So it is now, and it has heavy usage. You can see that in the web logs, and runs stably. So it's really a very solid, well-engineered piece of software now, and can really be used in batch mode without any problem.
So yeah, that's very important when you want to rely on this kind of services with web clients or pipelines, because there are several things to take into account. One is that the user at the other end of the browser, the internet browser, doesn't want to wait for more than 4 seconds, for example, for a, let's say, incident. You know, in the case of the analysis, I understand that it could be suitable for the user to wait for more than that, but we aim to give the user always a response in less than 4 seconds. And the other is, if we have a lot of people connecting their pipelines to the analysis service, we don't want people to have the pipelines broken because the server has gone down, and for the same reason we don't want our server to go down. So it's not only for one user, it's when you have several users connecting to you at the same time, you want to be able to handle it and to send back the result to everyone. Okay, so well, now the Tomcat has already started. I'm going to make this window a little bit bigger to have a little bit more space. Okay, so this is how the pathway, the firewalls would look like at the beginning. So the next version of the pathway portal is, no, they can only say this one. So you are now on your screen is the firewalls. This is this. Okay, so this is the firewall. So as you can see, well, as we can see, when we just keep moving the mouse on top of this, the different pathways that we are selecting appear on top of the display. And if we are moving the mouse here, so the different names in the diagram are popping up. So we can, for example, select a pathway here in the firewall, and once this pathway is selected, we see the information here in the overview, and we have seen that in the hierarchy, it has just opened all the way down to the selected pathway. We can go, for example, we can select another pathway. I'm going to select now the MAP kinase activation in ERR cascade, and once that is done, then I will focus in this pathway, and we have a detailed view of what's going on here. So now, for example, if we go to, I want to find in the signal translation. Okay. In the signal translation, we have one pathway that is the valve map. So one of the nice things that the firewalls is showing us is when we click valve map, we see that valve map is related with a lot of pathways at the same time, and it's part of 3 different major let's say benefits or top-level pathways in Reactome, that is the signal translation and the development path. Okay, now I'm gonna analyze some example data, like for example these gene names from liver that we have in the example. And once the data is analyzed, now I'm sorry, I have to remove this manually and reload because this is something that I haven't finished yet. So it needs to reload to show the firewalls again. And what we see here is the result of this over-representation overlaid directly on top of the firewall. So if we zoom in, for example, to this area, then we see that the yellow ones, so for example the orange ones, okay, we see that these ones have better significance than these brown ones here. Okay, then when we select one pathway, we see, for example, the level of significance here. And okay, because I think we are almost, I have to finish the presentation, it's 4:47. What I wanted to show you at the beginning, but I'm quickly going to show that, is the firewalls for different species. So we have Homo sapiens because the other I already showed. So we do this through Arabidopsis. Okay, what we see is that the firewalls gets cropped, and then only we have part of the pathway. So if we keep doing the same, if we go to Gallus, this is a special case because in Gallus we also have curated data. So this new curated data, the firewalls represents it in this code here. And then we have other species like, I can do this, like the Mycobacterium tuberculosis that doesn't have any pathway with the human, and then has a completely different kind.
There's one question in the chat box. Can you see it? Well, I'll just read it. Just will read it out.
Okay. As I understand it, the input for reactome pathway analysis is protein and gene ID list. Any plan to incorporate quantitative proteomics data into this analysis in order to sort of weight each molecule in the pathway?
So the question is, can we include quantitative data in the pathway analysis? And the answer is yes. You can already now provide a tab-limited input with, for example, a time series of proteomics, so protein expression or gene expression data, and then this can be run through the analysis, but and it can, the data and the quantitative values can then also be visualized on the pathway, on the individual proteins or chemical entities, but we are not yet doing a very sophisticated analysis of the quantitative data. So we are simply, I think, averaging across all the time series, time points for each of the proteins. And there you could do more sophisticated analysis, and we are open to suggestions, and this is one of the areas where we can do that further.
Okay, thanks, Henning.
I think we've used up our time, right?
Yeah. Are there any last questions before we move on to the second presentation? All right, so we'll continue this discussion next week. If anyone has any questions for Reactome, we'll continue these discussions during our open discussion period next week, or even after this meeting. All right, so thank you. Thank you, Antonio and Henning.
Thank you all.
For that presentation. All right, now we're going to move on to our second presentation. All right, so our second presentation will be presented by Chunlei Wu. Chunlei has a PhD in biomathematics and biostatistics from University of Texas. He has worked at TSRI for 4 years and is currently an associate professor of molecular medicine. He will be presenting about MyGene.info, which provides up-to-date, high-performance web services for gene-centric annotations. It also is a framework to host other types of large-scale annotation data aggregated from the community. Without further ado, allow me to introduce Chunlei Wu. Go ahead.
All right. Yeah, thank you. This is Chunlei Wu. I'm going to present this project developed in our group called MyGene.info. And just like Vincent introduced this, it does like one simple task. It's providing bioinformatician of high-performance, reliable web service for the query the gene annotation data. And it also has like a framework so that we can apply this same framework to other annotation data. And in this presentation, I'm going to touch on another related project called MyVariant.info. That's specifically for the variant annotation query. Okay, so Okay, so as a bioinformatician, we realized like we are spend a lot of time doing the data wrangling. For example, if we want to get the gene annotation data, we oftentimes find ourselves we need to download a flat file from different resources, and do the parsing, and then convert it to something usable in our application or analysis pipeline, convert to the internal gene object. And those data will be updated in different frequency, and then over the time we need to manage how to do the upgrading and how to store the data in the local database, do all the maintenance. And all these tasks pretty much like all bioinformaticians are repeated this same effort when there's occasionally the flat file format change, so we almost like everybody need to feel like, oh my god, this format has changed, and everybody need to modify their data parser and pretty much do the same thing. So we find that this is like a so repetitive effort, so that make us think maybe we can create something like a web service so that we can abstract this type of task out of the bioinformatician, so that bioinformatician can focus on the real like a downstream data analysis part. So that's why we created this MyGene.info project specifically for the gene annotation data. So to make a web service suitable for for the real-time applications, we think like the speed, the performance is really important, because you don't want to rely on web service which probably like a response in more than like 2 seconds. You want to instantaneously like get a response from the web service. It must be fast, and it must be very stable, and it must be like always on. And as the data provided, it should be up to date, so that bioinformatician don't need to worry about updating the data, worry about the like outdated gene annotation data. And of course, when the web service provider become more and more popular, it must be scalable and extensible. Okay, so here's what we are doing with for this problem in MyGene.info. So we first we created some like a parser to transform the data from different resources like NCBI entrance, ensemble, UniPro, and all the different resources, converted the data into an object we represent it as like a JSON object. Oops. Like a JSON object. Maybe use this as a dummy example. So for example, like from entrance we have like a gene 1017, that's just the gene ID. From ensemble maybe we got like ensemble ID. From UniPro we got a UniPro ID. Then we can merge them together, become like this object. That's everything we know. That's not everything. Like this is like something we know for the gene 1017, which is like a CDK2. And then we can pass to the query engine to do the indexing. That's why we then we have like when we say like we want the information about a gene CDK2, then we know this object is everything about the gene CDK2. And this shows you a more realistic example of a gene object. That's like we have the key. Key here is using the gene ID. When gene ID, this is like an NCBI gene ID. When NCBI gene ID is not available, we using the ensemble gene ID here as a gene key. And then you can see that all the rest of the information related to the same gene are aggregated together here. Here we're using a JSON format so that we can not only host the information like a symbol CDK2, just like a simple key value pair. We can also using the like a structure, like for example, if there's like an array of transcript ID for the ref-seq, then we can use an array, or even like we can use a more hierarchical structure to represent the annotation data. Oftentimes the gene annotation is a hierarchical structure. It's very not like uniformly represented as like a simple key value pair. We need some kind of a hierarchical structure to represent the annotation data. So this is a simple gene object represented as a so-called like a document format, and this is probably like a more full example for this CDK2 genes. As you can see here, we basically dump everything about the same gene into the same object. So you can see all kinds of like information about the same gene here, and I actually collapsed all this information. If I expanded this object, that can be like a very long object. But that's everything we aggregated for the same gene. So now we have the gene information about all this merged information about it for each gene, so that we can pass to a powerful query engine, do the indexing, so that we can query for, not based on the gene ID, we can query for like basically every field, every subfield, every value in this big project. So in the real in the production deployment, we actually need to deploy a cluster of a query engine here in order to achieve the really fast response for the user queries. And when user queries those, our query engines, it actually doesn't depend on all the backend data hub here. Data hub here is doing all this number crunching, preparing all the data. Once everything is ready, we then send it to the query instance, and that's actually the host is the indexed data, so that we can handle all the user queries. Okay, so for the end user, this is what we provide for the user. So really the API provided from MyGene.info is really simple. Even though like it actually hosts all species, you just need to have two endpoints. One is what we call like a query service. You just take any query term, any like symbol, any full text query, any identifier. It will return the matched gene hits. And another one is like called gene service, which is more or less like a convenience way of doing the previous query, because normally if you know the gene ID, either NCBI gene ID or ensemble gene ID, then you can get back the whole gene annotation object directly. That's something you can do if you know the gene ID. And those API is very simple. You don't need to sign up. There's no API key. You can try the live API here and do some tests from our website, and you can easily use it in your application. And also, you can know more from the documentation. Okay, so here this slide summarizes all the data coverage from the MyGene.info. As I mentioned earlier, it actually supports all species with the gene annotation. At this point, right now, we have more than 15 species. Okay, now it seems like better. I hear some feedback. Okay, so I repeat here. So the data coverage in MyGene.info here is summarized in this slide. It supports all species with the gene annotation. That's like more than 15K species for more than like 18 million of all genes. So that means like MyGene.info service can support all kinds of applications from human, mouse, rat, all these common species, to the even like a microbian study with all the different bacteria species. And we currently include more than 50 annotation fields for the genes, and we are constantly expanding our annotation coverage. And the data provided from MyGene.info is always up to date. We keep a very tight update schedule, which is weekly updated. So that data will always in sync with all the all these data sources we load the data from. And for the more information about the current data coverage, you can easily get this information from this metadata service. Okay, so for the MyGene.info query service, actually support the very flexible query interface. Here's I list all kinds of query you can do. I just like almost like everything you can think about it. You can do with all this like a simple queue parameter with very flexible query syntax. And I want to highlight one is here is called like a fielded query, because we have in MyGene.info, each gene object is aggregated in a big object. So you might want to like query for like a specific field. For example, you want to query a symbol, so that you can restrict your query to a symbol using just like a symbol. And notice this like symbol here, you can also replace it by any field that you can see from that big gene object. It supports like a dot notation if you need to restrict your query to some deeper structure in this query. For example, you want to do like maybe reporter.u133a and comma then your like a reporter ID from the U138 U138 chip can be used for the query. And another one I want to mention is this is like a return field query. This filter. That means when you do any query with MyGene.info, you don't really need to get back the whole gene object. You can supply a parameter called field. That way you can filter the information returned to your application. And like you can do all this symbol, homology. You also support is like dot notation. For example, you need like a reporter.u133a chip identifier return as in your result. Yeah, there's all you can do the batch query. There's also other more advanced feature like faceting, paging, user filters. It's also supported, and more information can be checked from our documentation. Yeah, and also as I mentioned, we really put the performance as a very, very important focus for MyGene.info web service. This shows a snapshot of the Google Analytics. Shows how many usage for our web service. On average, we have serving about 1,000 requests per day, but in our stress test we did in current setup. We can handle for like 5,000 concurrent, more than 5,000 concurrent users for 10K requests per minute. That's really like a very high capacity we can provide for the research community. And if it's needed, we can pretty much just increase the number of cluster in our query instance so that we can handle the much bigger requests. Okay, so this actually shows you the another usage stats that show the 1 month period. On average we got like a 2 to 3 million requests per month. But sometimes we just got like a really big hit, like this tweet I recently tweeted about it. It's during Christmas, someone apparently just hit our web service like crazy, and he's apparently still working during the Christmas. Like we got like 3 days continuously for the 5 million hits per day. Yeah, we are glad that like our system actually held up and everything is okay, and we can provide that that amount of web service request to our user. And yeah, this is a very nice stress test, really real stress test from our user. Okay, so this slide also shows a real-world usage. In real-world usage, all the web service hit MyGene and hit to the MyGene.info server. Actually, most of them they finished really quick. They finished within like 20 milliseconds, basically. This is the query service. This is for the gene service. They're response very quick in the real-world use case. We also provided the MyGene.py Python package for accessing MyGene web service, and I won't go into the details about this. And also we have the MyGene.R package. We just released end of the last month together with Biconductor version 3.0. We also have this third-party package called MyGene.info autocomplete widget. This is just like a Java widget. So like you can grab your code use it in your web application. You don't need to set up your backend data service, and then immediately you have auto-completed gene input widget in your web application. You can customize it. You can add as a callback function to this widget. Actually, we think this might be a good candidate to fit in the project like ByJS project. So far I have talked about the MyGene.info as like a web service, so that an end user can directly access the up-to-date gene annotation data, make queries, and use it in the live application or the analysis pipeline. And then for the data provider, we think that this MyGene.info framework is also an easy way for them to contribute to publish the annotation data. For example, you don't really, this is an example here. For example, from our this BD2K project, maybe some analysis pipeline come out as a result is called a cardiovascular disease associations. Maybe from this paper, the curator gathers the data about this this gene CDK2 has an impact on the impact of the RB phosphorylation in this heart smooth cell tissue. Then this is like information. As long as you have the parser to creating the data structure like this with the gene ID, and the rest of the information is totally up to the data provider. You can have your own data structure. Then we can put into MyGene.info machinery, merge with other existing annotations, and immediately for the data contributor, you have the high performance web service to access your data, and together with all the other annotations, you can do the flexible query interface. You don't really need to worry about all this maintaining database or developing a really high performance scalable web service. All this thing, we will take care of all this for you. This is a more like a realistic data importer from the contributor from the community. For example, this is a simple data loader to load the pharma GKB ID mapped to the gene, each gene. So you can, as long as you have this something called like a loaded gene doc, that function basically loaded the information from the flat file, and then you have the get mapping basically returns the type of the data you contributed to the MyGene.info. Then you can integrate it into the MyGene.info platform. Okay, so we developed this MyGene.info initially for the gene annotation purpose, but really the framework we developed is neutral to any type of gene, any type of biological annotations. For example, we can extend it to other entities like a gene, like a variants, a disease, metabolites, and pathways. All the bioinformatics, biological knowledge, I think they are like central around this, all these biological entities, and they have the relationship between them. But all the annotation, they have like some kind of ID, and then have all the annotation associated to this identities associated to this each entities, so that we can use in the MyGene.info like framework to handle all the annotations for these entities. In fact, we already launched new application called MyVariant.info to handle the variant annotation. In the variant annotation world, that annotation is the fragmentation is much worse than the gene annotation. Right now, we don't really know. We don't really have a consistent variant ID to represent each variant identified from the data. So, so in our case, in MyVariant.info case, we decided to using something called HGVS name as an ID. It's not 100% perfect, but it will cover the most of the case. I would say like 99% of the use cases. Just like I showed here, for in MyVariant.info, we have the ID as the HGVS name like this, and we have like annotation associated to this variant. For example, this is from dbSNP. All the information we loaded from dbSNP comes to in this field. And this is another example. I basically collapsed all the information, but you can see we can aggregate all the information from Cosmic, dbSNP, dbSNP, all these resources, and all the variants annotation about this same variants can be aggregated together. Then we can serve to the end user with our query engine. And this slide just shows all different common type of the HGVS name we can represent, not just the single nucleotide SNP and single nucleotide substitution. We can represent it in myvariant.info. We can actually represent all this common type of variants identified from the data. Okay, so here's the stats for the myvariant.info. Right now we have loaded about 116 million variants and aggregated annotation from all this over like 10 data resources, and there's also a few, and there's like probably a dozen of them currently in our pipeline, and we want to aggregate it to the myvariant.info system. And I want to highlight here is like we recently loaded Welderly data collaborating with Ali from the STSI, and that's about like 20 million of variants directly from the Welderly project. Now you can query all this variant data using the myvariant.info interface. Okay, so this slide I want to summarize how the MyGene.info, myvariant.info project might be relevant to this BD2K project. And this is probably like overly simplified schema, but I want to summarize is that MyGene.info, myvariant.info can be a data backend provided to other BD2K applications. For example, maybe like a Reactome, if want to display some gene information in the pathway view. Maybe you can, there's like some JavaScript widget you can like develop easily so that grab the data from MyGene.info so overlay some gene annotation, gene information box in a Reactome interface. That's possible. And also for the annotation data we might get from other BD2K analysis or applications. If they are gene-centric or variant-centric, or maybe we can develop some other entity-centric annotation query engine, like mysomething.info, so that the data can be imported into this system, so that we have we really have a high-performance public API provided to end user and also to the other BD2K applications. And that's all my talk, and this is our team behind MyGene.info and myvariant.info. This project led by Andrew and me, and this is Adam and Ginger are the team member in this project. We also work with our team of outside developers, Xuefeng, Tiebao, and Wei. So yeah, that's all my talk today. I'm happy to take any questions.
This is Pei Pei. Thanks for a really informative presentation. I've just been so impressed. So could you and Andrew give us a bit more of education on how would MyGene.info would actually relate to the GeneWiki project, or this is a project in parallel? I definitely see the very obvious connections on the biomedical side of it, but with respect to web organization and database organization, what are the connections of the different projects?
Mm-hmm. Yeah. Gene Wiki project and MyGene.info project is like a parallel project in our group, and also
it's like a sister project.
Yeah, they are also connected. For example, the some like Gene Wiki is basically providing the non-structured, unstructured text-based annotation for the gene, right? So, and also you can notice there's a box on the side of each Gene Wiki page that's providing all the structured information about that gene. That information actually is populated from the MyGene.info service. And
I see. So there is a backbone connection. How are you achieving that?
Oh, that's
How's that program done?
Yeah. So in terms of like implementation, that's actually there's first the Gene Wiki actually has API, so that we can write a script to basically send the information using a script to Gene Wiki, so to populate that gene information info box. Yeah, and that can be done by the Gene Wiki not Gene Wiki, like a Wikipedia API. Yes.
Okay, so that's very helpful information. So the Gene Wiki or the Wiki API, how do we access that? So for example, if Reactome is to connect to Gene Wiki and KabaKB is to connect to Gene Wiki, should we follow your examples of how you connected MyGene.info to Gene Wiki? Or what are the technical information or examples we could follow? Can you direct us to that?
Yeah, one thing we can do is if depends on type of data we got from Reactome, from other project in this other applications in this project. We can, if that fits into the like MyGene.info or MyVariant.info, we can pull the data into MyGene.info, for example, and then we already have the script, have the code to populate the data from MyGene.info and send it to Gene Wiki. That's totally doable.
That's okay. So that's definitely one pathway to do it. Is we need to go through MyGene.info then to Gene Wiki. But what are the ways that there will be a direct connection between Reactome to Gene Wiki and between KabaKB to Gene Wiki? We definitely could make a stop at MyGene.info. That's definitely going to enhance the annotation and also structure of the data. But does Gene Wiki have a place where we could negotiate collaboration? How did MyGene.info negotiate that collaboration with Gene Wiki?
I believe like Gene Wiki is hosted in the Wikipedia, right? So Wikipedia is the public API. So you register. Once you register, you can have access to this API, and so that you can write the program to basically changes how the wiki page looks like, changes the content in this page. Yeah.
Okay, that's very helpful, because one of our collaboration projects with Anju and you at TSRI is to actually work on how the cardiovascular genes will be better annotated. So maybe it will be helpful that we could work on also a parallel technical project to see if we could connect to both MyGene.info and to GeneWiki. Should we contact you for technical details on this?
Yeah, so
Would that
like
be okay?
also specifically for the GeneWiki project, I think Ben is the most like the lead person in our group. So yeah, he will be most familiar with the GeneWiki site, and for my Gene.info site, that's definitely like I can help. Yeah, but together basically, yeah, we can have a meeting between Andrew, me, and Ben with your group, and we have to
yeah. Okay, that sounds wonderful.
Yeah, definitely doable, I think. Yeah.
Thank you.
And I imagine the data incorporated from CopperDB, Reactome, that will be something like a structured data, right? Annotated data, not like free text, I imagine, right?
I can't speak to Reactome. Henning is the head of that project. With respect to CopperKB, at this moment, all data in CopperKB are structured.
Yeah.
But we are in the process of opening up to non-structured data.
I see.
And so we'll see how that would be moving forward.
Yeah, sounds very.
Maybe Henning, I'm not sure if they're still there. They could comment on Reactome, but it is very late for the CI people. Okay.
So Reactome is a structured resource, and you can, I would imagine, if you want to include content from Reactome, probably more into what you said, my pathway or my path, the future project, but there might also be some for MyGene. Yes, you would just pass the structured data and include it. In the other way around, where we now have any reference to a UniProt protein, we have a whole bunch of references to external sources like UniProt, Ensemble, and so on, and we could include one to MyGene.info. So I think that is really just, it might be work, but there's no major, it's not a big thing to connect between the two.
Yeah, sounds great, yeah.
Chunlei, you have several questions in the chat box. Can you see those?
Oh, okay. Let me check that. Okay, so let me from the beginning.
I think, Antonio, you should probably explain a bit what you did for your, so the first question I think came from Antonio, right?
Yeah, there's a bunch of questions. Let me scroll up to the first one.
There's more, yeah.
I think the first one is from Brian.
Yeah.
Yes.
If one kind of identifier is not unique to a single gene, could the algorithm deal with the redundancy? Yes, of course. For example, if you have, say, like one gene, maybe now is the time to do some like a real demo. It's easier. So MyGene.info, that's our website. Yeah, you can try this like a live API if you want. This is gene, this is query. Also, let me see. To answer your question, is there like one kind of identifier is not unique to a gene? Maybe say, let's do a query, for example.
Right, that's Antonio.
There's like a PFAM ID. For example, let me show this gene ID. For example, we have maybe this like PERSite. This is not unique for a gene. If we do a query like this, it will return all gene assigned to this PERSite ID. Does that answer your question?
Yes. Yes. Thank you.
Yeah, you can. Yeah, it's like you can see how many hits. This is like 200 hits here, and this is actually obviously like return just a 10. you can do all the paging or this thing to get back all the information. Yeah. By default, it will just show the symbol ID, but you can easily do like fields. You can say like if all, then everything will return for this particular gene. Yeah. Okay, so let's move on to the next question. So next question from Antonio. MyGene.info query P10.
Yes, because P10 in the Reactome case is also associated to different entities in the diagram, and then I was wondering if I want to connect to P10 to just see all the different resources that you have interconnected, because when you do this query now, this is the result for this. Yeah. So now we see that the same. We have several hits. Okay. Then, for example, for the second query, I've chosen the interest gene, the first field, so I understood that that is an identifier, and then I just use the gene slash and this number, and then it gave me another result, and I was trying to interpret this second result.
So I wasn't. Yeah, I'm not sure I follow exactly. So you did this query P10.
Yeah. And now we can take
what?
The first entry.
Yeah.
And that is an identifier, isn't it? 5728.
5728. Yes.
So you copy that, and then in the URL instead of query, you go to gene, and then slash, and then this number. Okay. I was trying to interpret what's the result in here. I just wondering, because now I've seen that you have this genomic, then you have the RNA, and then you have the protein, but I don't identify any. Yeah, there is one uniprot protein.
Yeah.
But can you explain a little bit?
Are you asking is this the right workflow? Or?
Yes, a workflow. Yeah, the way to query your results, or if this or there is a simple way, or I don't know.
Yeah, like and actually, if you want something back, you can really just do it from here. For example, because here you can not just return all this gene ID. You can return anything else. So for example, you want like exons. You can just do this exons, right? So it will return directly from this query. You don't really need to go to the gene query to get back the gene information. Also, you can specify, say, like a species.
If I wanted to, for example, see all the uniprot identifiers associated to the P10 gene.
Or what? I'm not very clear.
Okay.
What kind of data you want?
Instead of P10, let's use UNC5B.
UNC
5B.
5B. Yeah.
Yeah, and now I would like to see the uniprot identifiers that are related to the gene.
Yeah, uniprot. You got that. And assume you want a human, right?
If there is more than one, then I get, instead of an object in the uniprot, I get a list of objects. Right?
If it's more than one, then it will be a list. Yeah, it will be an array. Yeah.
Okay. So I don't need the second query is not needed. I can filter by this one.
Yes. Mm-hmm. Yeah, you probably want to check like if it's like an array or it's array or it's just single value. Yeah. Yeah. Mm-hmm. Okay. So does that answer your question?
Oh, yes.
Okay, great. So that covers your next one, I guess. Okay. Yeah. So next question from yes. The merging algorithm is working in a cluster environment, or is it pre-computed? Yeah, this merging actually is just happen in our so-called like a data hub. And yeah, let me go to the slides. Yeah, merging actually just happen on this data hub. This is like just one server. And every time we, this is actually there's like a program to monitor all the data source once there's update, and put down locally and do the parsing and convert it to so-called like a JSON format or document format, then do the merging, and once every merging, actually there's one detail I haven't covered here is that once all the merging actually does some kind of like a diff with last week's version. So every time when there's an update, actually we send just not all data, we just send the difference of the annotation, send it to all this cluster of a query instance. So every week we pretty much just send like maybe 20, 30 megabyte compressed data. We don't really send the whole like a couple gig of all merged data. Yeah. So yeah, it is pre-computing, and it's independent of the actual query instance, so that when we do all this number crunching, it doesn't affect the actual public query instance. Yeah. Okay. And next question: do you expect the users to concatenate this?
That's sorted.
That's what, yeah,
That's sorted.
Yeah, I just wrote for messages, but all was the same. All of them were the same question. So
oh, okay. All right, great. For clarification, do you store the data from the database? Do you store the data from the database and store it in your database to be indexed? So what type of database do you use? Yeah, we grab all the data from other database. There's probably like more than about 10 databases right now. We take all this data. Most are the flat file, but some are just other like a database query or yeah. We do the parsing and store it in our local database. And this merging, when this merging done, all the data stored in MongoDB. And then once in MongoDB, we do the diff computation between the two merged database, current one and the last week one, then compute the difference. And then the query engine is actually Elasticsearch based, so that allow us to build like a large query cluster to provide all this powerful query interface to the end user. Yeah. Does that answer your question, Vincent?
Yeah, that's perfect. Thank you.
Great. And next one, technical question. What technology do you use to create the API? Yeah, well, Data Hub, that's, overall, our applications are written in Python. And Data Hub, that's a lot of Python code manipulating the MongoDB. And query instance is a Python code, and the web service is actually based on something called Tornado. And that's really a high so-called like a non-blocking web framework, so that it can handle really large concurrent user queries. So that's yeah, and all the indexing is done by Elasticsearch. Yeah.
Yeah. Thank you.
Cool. All right. Any other questions?
No, I've got an additional question.
Sure.
How many, you showed on one slide, I think 5,000 concurrent users. That's a very high number. So how many distinct users per month do you have?
Every month we check I actually just checked before, yesterday when I preparing these slides. Every month we check about 2,000 to 3,000 unique IPs.
But then I don't understand why you have 5,000 active visitors. Is this? I thought this was on one moment, in one moment of the slide. So how can you then?
Yeah. This is like something we record from the Google Analytics. We feel like very excited. Sometimes just the numbers goes really high. And actually, in my past presentation, I actually like to show the live number. Say, I don't know, with real time. Yeah, real time. I don't know. Let's see how many people. Whoa, that's actually quite a bit. Yeah, that's like show like a 600 active user right now making the query. Yeah.
But what I don't understand, in the previous slide, you show 5,000 active users on site. But then you say you have only 2,000 to 3,000 distinct IP addresses per month. So how can it be less per month than you had, or was this just one exciting month, but the average month is lower? I don't quite understand.
Oh, this IP really change very a lot. So this particular month maybe have more like unique IPs, and then like the last month I just checked the January, and we have like 2,500 unique IPs.
Okay, so this was a good month basically, because I would guess if I have 5,000 active visitors at a given moment, then I would expect the number for that month to be somewhere like 10,000, 20,000 in distinct IPs. So that's why and that would be really high. That's why I'm surprised.
Active user. Yeah, I honestly, I really haven't dig into how Google calculated this. I'm not sure if that's related to directly related to it probably related. I don't know if that's just like a linear relationship with a unique IP. Yeah, I'm just not sure. Yeah.
Okay.
Yeah.
You're using the Google Analytics from the to the client side. I don't think so. So it's something that you have to include in the server side just to report to the Google Analytics, isn't it?
Yes. There's a package called a PyGA or something does that. Yeah, you just it's almost like a client side. You just do server side whenever there's a request in.
What's happening is that someone is hammering your server, but the Google Analytics doesn't say, okay, this guy is connected and suddenly is disconnected. So it keeps the connection it keeps the user connected for a while. So if someone is hammering, then you are counting right now so many users, but it actually is what happened in the last few minutes altogether.
Yeah. Yeah, I believe like my imagination was like Google probably like treated this even like the same request from the like a different request from the same IP will probably treat it as active like a different request because that's like from server side. That's from server side, so there's no like a cookie or anything can track like a session. There's no concept of a session. That I guess that's probably why. Yeah.
Okay. I've got one other content question.
Sure.
Often the challenge is something like you have list of mixed identifiers, whatever, from some bioinformatics workflow, and the challenge is not to get as much out for each identifier as possible. But the challenge is to map all of these to one common reference system and to choose the best reference system. So basically for things where, let's say there's the same in your bunch of input identifiers, you have data for, let's say, one sequence which is in SwissProt, and then there's the same protein identifier for the same sequence, but it's in Tremble because it hasn't been merged to SwissProt yet. So, but you really want to have to know that, I have two references to this SwissProt entry and give me only that one back. So basically map mixed identifiers to one good reference system. I think, what you have now on the screen is the answer, but I just wanted to check back with you, is that a functionality you explicitly support?
Yeah, so I want to make sure I understand it correctly. So you mentioned like for the uniprobe case, that when there's two identifiers, and they are matched, basically like I think we don't do any like a mapping. We don't make any call for, let's say, like this SwissProt ID should map to this gene. We, when we load the data from uniprobe, we take the uniprobe's mapping to gene ID. Because we don't want to put ourselves in a position to make that call, say like which one should assign to this gene when there's like an ambiguity. We pretty much leave to the original data source to make that call. I'm not sure if I answer your question.
Okay, I'll try to rephrase on the gene level. So I have a list of something which fell out of my my experiment, whatever it is, and I have half of the genes are ensemble identifier, and the other half of them are, let's say, entree genes, entree gene identifiers.
Yes.
So do you support the workflow of map all of these to ensemble and at the same time tell me, let's say, all those entree ones for which you didn't find a mapping? But I have unified result set, because that's a very frequent challenge. And what you did here was selecting UniProt might already go, or SwissProt might already go into this direction, but did you think about this use case, and are you supporting that?
Yes. Actually, we do support that. When you make a query, actually, let me show you. Let me show you this. We actually made a Python notebook show like the ID mapping case. Here is the tutorial we wrote. Let's see. So when you make a query, you can actually, when say like your input is a mix of identifiers, mix of your ensemble ID, entrance genes. Sometimes if it's sometimes like it's in array annotation, if it doesn't map to either of the gene ID system, it's probably gonna be like a sequence ID. So in that case, when you do the mapping, that here's like I show the MyGene Python client. So when you do the mapping, let me show you an example. Scope, right? Here's the example. When you have the input, this is like not just one type of ID. You have mixed ID, right? So you specify something called scope. So it tells your input ID there's like a symbol, mix of the symbol, reporter, accession. So then there's a field you say like I just want to return entrance gene and UniProt data. So now you can make this query. It makes basically the one batch query. It will do the query, tell you, okay, this one doesn't find the duplicates. Like one ID map to two genes. And this one find like no hits. And this result basically will come up with this. As long as any of those, regardless their type of ID, you will get it. If there's a map to the genes, you will get back the information you specified from the field parameter. Like this.
Excellent. Okay, that answers the question. That's very good and useful. Thank you. That's something which comes up quite often.
Yes, absolutely. We also find that this is oftentimes it's very annoying to do all this kind of mapping. Great. Any other questions?
Okay. Any last questions?
Last orders, please. Before we go to the pub. You still have to work.
I'm jealous.
It's
too early for the pub here.
If there aren't any more questions, we'll conclude the conference here. We'll conclude the presentations here. You guys are free to go, but Brian and I will be here until 10 to answer any questions. But thank you for coming. Have a good weekend.
Thank you, everyone.
All right. Thank you all.
Same to you.
Thank you, everyone.
Thank you, Brian and Vincent.
Appreciate it.
Thank you. you.